Hello everyone, my name is Semal Kunal Prasad and I'm a 5th year veterinary science student at the Fiji National University, and today I'll be speaking on the topic in vitro embryo production in cattle, sheep and swine. So what is in vitro embryo production? The word in vitro is derived from a Latin term meaning in glass, and it is one of the advanced techniques in the field of biotechnology in respect to animal production, whereby the embryo is produced outside the animal's body, and it is usually carried out in a laboratory which contains proper facilities to do so. And in this process, three stages are mimicked, that is oocyte maturation, the process of fertilization, and initial embryonic development. So this process is carried out mostly in large farm animals compared to small companion animals, and there are various selection criteria in order to choose which dame's ovary will be used as a sample to generate. Now selection can be based on examining the pedigree, that is looking at the ancestral line of various animals, or by the appearance, their phenotypic and genetic characteristics, because the main objective or the main aim of this procedure is to produce superior offsprings compared to their parents. So in this process, the undesirable traits are eliminated as much as possible. Now coming to the process, so in vitro embryos are obtained by a series of integrated and efficient manipulations of follicular oocytes, and there are basically four technical steps involved. The first one is oocyte collection. The second one is in vitro maturation of immature oocytes. The third one is in vitro fertilization, and lastly is in vitro culture. Now oocytes are female germ cells which can be obtained from ovaries, and samples of ovaries can be obtained from slaughterhouses or abattoir, but it is more desirable to obtain fresh samples, that is from live animals. So there has been various advancements and refinements in this field. So there is a process known as follicular aspiration by laparoscopy, also known as laparoscopic ovum pickup, and it is the most desirable technique to obtain fresh ovary samples from. Now we come into the second technical step, which is the growth and maturation of immature oocytes. Now the growth of ovarian follicle is really important, and there are two main phases: the slow growth phase and the second one being the slow growth phase lasts for only about 30 days from antrum formation, which reaches a size of about 300 micrometers, and this is a very critical stage for the development of oocyte capacity, since this is the time where the oocyte reaches its final size and competence. And if you compare it to the fast growth stage, it lasts only about 5 to 7 days from follicular antrum detection and reaches a size of about. To add on, we must also consider hormonal factors. Hormones such as follicle stimulating hormone, luteinizing hormone, and gonadotrophin releasing hormone influence the growth of follicle, thus the oocyte. Now, once desirable samples have been acquired, now comes the most crucial part, which is in vitro fertilization. Now, since we are going against the natural process, we require fresh semen samples or cryopreserved semen. It is really important to note that seminal plasma or extenders contain components that serve as decapacitation factors, and this has to be removed before it is incubated with the oocyte, because this can affect the process of fertilization. So this is achieved by simple centrifugation with a centrifuge, or there is another process known as swim up, where the sperm cells, which are highly motile, are allowed to swim up to the surface. Now the ideal temperature for cell culture incubator is 38.8 degrees Celsius, and it should have a humidified atmosphere of 5% CO2, and for a period of 18 to 24. Now we come to the advantages and disadvantages of in vitro embryo production. So if you look at the advantages, there is quite a few. Firstly, you can produce large number of offsprings in a year, and you can eliminate undesirable traits and create superior offsprings, and also you do not require large number of male animals because you require only semen samples. But if you look at the limitations or the disadvantages, there are quite a few, as you require a lot of practical skill and knowledge to carry out this procedure, and it can be costly too, because you require proper laboratory facilities, proper equipments to attain maximum results. Also, if you look at samples, it can be really difficult at times to obtain proper samples to carry out this process. And last but not the least, at times the process of in vitro fertilization can fail due to certain minor mistakes which can result in polyspermy. So this is all I have for you today. I hope you enjoyed the video, and peace out.